So what I'm going to do is spend a little bit of time explaining about mitochondria and mitochondrial disease and talking a little bit about research. I'm going to talk a little bit about some of the research that we're doing on IVF techniques. My colleague, Professor Chinnery, is going to talk a little bit about using new tablets to treat mitochondrial disease, and Dr. Gorman's going to talk about some of the studies that are ongoing. So what are mitochondria and what are mitochondrial diseases? And I suppose many of you know this already, but I just thought I'd take you through it because I think this is relevant to all of the kind of research that we're doing. So we often describe, and people will often hear me talk about mitochondria as being the batteries that are present in our bodies. And that's a very good example because batteries provide energy. Your torch wouldn't work. These sophisticated cameras wouldn't work without batteries. And we need those batteries to work. The mitochondria are the batteries that are present in every cell in our body. So our nerve cells, our muscle cells, our heart cells. And what happens when the batteries start to fail? What happens when the mitochondria start to fail? Well, you can see that one of the things that happens is the tissues, your organs, start to fail and they just don't work as well. And of course, if things don't work, if the batteries don't work, then what happens is it can lead to heart problems. And many of you will know that we often checking people's hearts. Many of you will develop some neurological symptoms, maybe problems with your eyes, possibly a bit of unsteadiness. Some of you develop diabetes. And again, that's because the mitochondria aren't working properly there. And some people will develop muscle fatigue or weakness or pain. The other thing about mitochondrial disease, and it's a complex group of disorders, and I'm sure you've all experienced this, going to your doctors, and the doctor's not really being too sure what we're talking about. And it is a very complex group of disorders. And one of the reasons is that it's rare that people have exactly the same symptoms. And that means that patients present with different problems. And they present with different problems at different ages. And one of the reasons is that many different tissues are affected. And it's those combinations of tissues being affected which gives you the symptoms that you have. And I think this is one of the things that we're well aware of. And one of the reasons why we are so careful about the way in which we actually discuss things with you, and many of you will find that we go through a range of different systems when we're asking questions. So what causes the mitochondria to fail? Why does the batteries fail? Now this is where it's going to get a little bit complicated, but I've got to go through some of this detail. Otherwise, you won't understand some of the work that we're trying to do. Everybody around will have heard that what actually happens is that all of us have things called genes. And what those genes do is that they make things. And we all need, the way we are, the way we're formed, is due to those genes which have come down from our mother and father. Mitochondrial disease is when there is an error in the gene that then causes a problem in the mitochondria. So there is a genetic, it's something that is in the genes which causes the defect. Now, many of you, or some of you, will be in this room saying, well, look, people have said that the doctors have said, I've got a problem with my mitochondria, but they haven't been able to be precise. They haven't been able to say what sort of gene is wrong. Over the next 3 years, I think this will change dramatically. And certainly, a lot of the work that's being done in Professor Chinnery's lab is about finding new genes which cause mitochondrial disease. And one of the things that's really, I think, almost beyond belief is how the advances in genetics are changing what we're doing. these are just, there's Dr. McFarlane, Professor Taylor, Helen, and this one of the laboratory scientists called Helen. And with a donation from a charity, we've bought this little machine. It's tiny. You can see it size there. And you're kind of thinking, well, will that little machine make much difference? Well, that little machine can sequence 300 million bits of DNA per day, right? So you can see that the human genome is 3 billion bases, and that there are now machines that can do that in a couple of days, even less. So you can see that the revolution in genetics is going to mean that those of you that don't have a genetic diagnosis, in the next 3 to 5 years, everybody will. And it's a clear link between our clinical service, the work that Professor Taylor and his team do, and the research work that goes on in Professor Chinnery's lab. So you can see that we are trying to provide that better genetic advice for people. Now, this is where it gets a bit more complicated, and many of you will have heard me talk about this. So here's the mitochondria, and this is a kind of different representation of the mitochondria. But mitochondria are made from genes which come from both the mitochondrial DNA, which is present in the mitochondria, or from nuclear DNA. Right, so mitochondria contain their own DNA, a tiny little bit. Most of the DNA in our cells, the bits that come from our mum and dad and gives us all the characteristics, is present in the nucleus. So this bit of DNA is about 16,500 bases. This is individual bits. This is 3 billion bases. So this is much bigger than this. The thing that makes this more complicated is that some of the genes that are produced from the nucleus, the main DNA in the cell, controls the mitochondrial DNA. And this can lead to different sorts of problems within the mitochondria. So it is complicated. I'm not expecting you to remember all this, apart from the fact that some of the genes which make mitochondria are present in the mitochondria and some are in the nucleus. I'm just going to go back and just say, so now I'm going to concentrate on the DNA that's present in the mitochondria. So this is mitochondrial DNA that makes mitochondria. And the reason I'm going to concentrate on it is that this is quite a common cause of mitochondrial disease, and I need to explain where we're going with this. Okay. So some of you in the room will have heard that they have a problem with their mitochondrial DNA. And what you will have heard me say is that there is a possibility of you transmitting this to your children if you're a woman. And the reason is that we get all our mitochondria from our mothers. The sperm from the father carries the nuclear DNA, but the mitochondria don't get in or get destroyed. So my mitochondrial DNA comes from my mother. I didn't get any from my father. Catherine's mitochondrial DNA comes from a mother, and her son has her mitochondrial DNA. It's the only way it's passed down. Okay. The other thing I just want you to realize is that mitochondria, this is just a nerve cell, mitochondria are separate from the nucleus of the cell. Okay? So mitochondria are separate from the main bit of DNA in the cell, the bits that give us all the characteristics, the bit that make us us, the nucleus. The mitochondria are separated. And this is the basis of the technique that you may well have heard about and has been causing all the fuss in the press, because what we're proposing to do and what we've been doing experiments to do is to take mothers that carry mitochondrial DNA mutations. And this is something that we want to try and offer people the opportunity to have children that don't carry the bad mitochondrial DNA. And the basis of this is that you get a fertilized egg just after fertilization. So it's got a mixture of the mum and the dad, and it's got all the nuclear genes that make us, but have got unhealthy mitochondria. We get a donor egg, which has got the nucleus removed, but healthy mitochondria, and we move over the nuclear DNA into this egg, which has got healthy mitochondria, which means that there is a potential for babies to have normal nuclear and normal mitochondrial DNA. So those babies would not have mitochondrial disease, but would otherwise be completely as we would expect. Now, this is something that we have been doing. I'm just going to show you just what involves the sort of work that Lindsay and Laura have been doing. And what we've been doing is using, you can see this is a human egg, and you can see that these bits that we want to move, the nuclear DNA, are nicely, you can see them down a microscope. And what happens is that Lindsay and Laura are using microsurgery. So this is it's tiny. I'm trying to think what 100 microns is like. But it's like the very tip of a pin. But Lindsay and Laura look down a microscope and are able to manipulate it. And what they've been able to do here, out of this egg, is take out the nuclear DNA. This egg's had it removed, and what they've been able to do is then put this nuclear DNA back into another egg, got it to fuse, and this is a developing embryo. So this technique is feasible, it's possible, and it's possible because this is the first time it's ever been done on human eggs. Now, clearly, we think this has got great opportunity, and something that mothers who carry mitochondrial DNA mutations might want to consider as an option when they come to want to have children. It has and is very controversial. It's very controversial because it's a new IVF technique. It's very controversial because it would be correcting a genetic disease actually before in an egg. And it's very controversial because it would involve us working on human embryos. And that's something that some people feel very uncomfortable about. So this is all highly regulated, and you'll hear a lot about this, and we will keep you informed. It's highly regulated by the Human Fertilization and Embryology Authority. Coming out next month is what's called the Nuffield Council on Bioethics report on this. And I just want to thank everybody that wrote in about mitochondrial research to the Nuffield Council, because they actually had a tremendous response from patients. And most of the time, they don't get responses from patients. They get responses from the people who have major vested interests. They get responses from people who don't want to do things, not necessarily hearing the patient's voice. And I think the patient's voice is going to be absolutely crucial over the next few years. Some of you have also been involved in the fact that this has attracted an awful lot of publicity. And what we do need to do is give both sides to the argument. There's always somebody that's prepared to stand up and say, no, you should not do this. What we need are people who actually experience the problem and are prepared to discuss this and be prepared, in fairness, to actually say it actually in front of the media. Because if we don't, then people, as I say, there's always going to be people who are prepared to stand up. The other thing is, before this could become treatment, and one of the things that we're doing over the next, one of the things that Lindsay and Laura and Professor Herbert are doing at the moment is working out whether this technique is safe and effective. So if we can show it can be done, but is it safe? And what are your chances of becoming pregnant after being done? And that's the sort of thing that we're looking at now. But before we could use this as a treatment for fertility treatment, it has to go through the House of Parliament. And the House of Parliament have actually requested that this Human Fertilisation Embryology Authority actually do a public consultation. So the public consultation will involve talking with the general public about mitochondrial disease, which we know will be actually quite difficult, but they also want to hear people's views. And one of the things is that I would, again, be very grateful, we will keep letting people know, to actually give their views on this. We don't want, again, all the negative side of this, because I absolutely respect other people's views. I think, I don't have, I think people's views are rightly, some of people hold very strong views. I just think we need to have a balance of views here. And I think that one of the things is, whatever your view is, you've either somebody with mitochondrial disease, or you've a loved one, or you've got friends with mitochondrial disease, your view is important as well. So there is something there where people can contact. And there is some information, isn't there, Catherine, in the kind of anteroom where you're going to have lunch, where we can give you details. And I think it's a very important thing. And I'd just like, and I'm sorry, this is my last slide, and I'm sorry this one's really complicated, but I think it's very important that people get the message here. There are two techniques for doing this. The technique that we've been doing is just after fertilization. It's called pronuclear transfer. So it's just after the sperm has gone into the egg. There is another technique which involves just working with the unfertilized egg before fertilization. And what we're doing is trying to see which is the best technique. The point is that this technique is, well, the preliminary evidence is this technique's more difficult to do. It might not be as successful as this technique. What the government are trying to push is that we're only able to do this technique because these are what we call embryos, these are just eggs. And what we're saying is, don't tie our hands behind our back, let us use the best technique. And I think it's really important that we get the message out there, and I'm hoping that's going to be the response of the Nuffield Council on Bioethics. Because I think what we want to do is not offer you second best treatment. What we want to do is offer people the very best treatment. So that discussion about, and people can come and have a word with me about this, this is quite an easy way, because as with most things in governments, they'll take the compromise situation. And that could be seen as a compromise, but I don't think it's in the best interests of patients.